Everybody, thank you so much for joining us for the February 2025 iteration of the seminar on physical genomics. My name is Benjamin Keene here at Center for Physical Genomics at Northwestern University. We're really excited to welcome our invited speaker today. Professor Andrew Stevens is Assistant Professor of Biology at the University of Massachusetts Amherst, where he started his lab in 2020. Professor Stevens' research uses a combination of nuclear force measurements and cell biology to determine the mechanical force balance between the nucleus and the cytoskeleton, which controls nuclear shape, integrity, and function. This work is certainly familiar to us here at CPGE, and I'm sure to many of you as well. Professor Stevens, thank you so much for taking the time to share your work with us today. A quick note for our audience. We have a Q&A session at the end of the talk. So you can either ask your questions then or just type them into the chat box anytime, and we'll address them when we get to the Q&A. If you're interested in learning more about the research and training programs here at the Center for Physical Genomics, please visit us at physicalgenomics.northwestern.edu. You can also check out our growing video archive on YouTube or follow us on Twitter, X, or Instagram. At this point, Professor Stevens, I think we can turn the floor over to you and get started. Thank you so much again for doing this.
Thank you for having me. So yeah, thank you to the presenter, or thank you to the organizers for inviting me to give you a talk today. So my talk is going to be focused on this specific project: transcription inhibition suppresses nuclear blebbing and rupture independent of nuclear rigidity. So this paper is published, and I'm really excited to tell you about it, but I hope you stay and listen throughout it, because we talk a lot about our work leading up to this specific set of work, and then work we're doing to follow up on this. So the nucleus houses and protects the genome. Reading of said genome dictates cell function. I'm sure everyone in the crowd is very aware of this. So we're really interested in the nucleus. This nucleus highlighted here in blue holds 2 meters of DNA, which is the genome, and a 10 to 20 micron size nucleus. DNA and its associated proteins make up chromatin. What's interesting about the DNA is it's this information system where the reading of it from DNA to RNA to protein determines cell function. So all cells have the same genome, but they can have wildly different behaviors due to the reading of this genome. And a lot of people think of DNA and chromatin in this very specific context of being information. And what I'm going to tell you today is that chromatin actually has a physical component to it. So chromatin is actually a major mechanical component of the nucleus, maintaining its shape and function. And so we really think about it as this polymer resistive function for maintaining its own informational properties. And so we'll be talking about that today. So the nucleus must resist forces and maintain the proper spatial organization of the genome under these forces, right? And so reading of the genome can also be determined by where it's located. So stuff at the periphery is usually off and stuff on the interior is usually on. But if the nucleus is experiencing forces, how does it maintain this spatial positioning? Also, there's mechanotransduction, in which the cell and the nucleus experience forces. These forces from outside, whatever the surface of the cell is on, can be transduced through the cell, usually through the actin cytoskeleton, both microtubule and actin-based, into the nucleus through the link complex, into the lamins and the chromatin, right? And so not only does the nucleus need to resist these forces, the chromatin need to resist these forces, it needs to respond to them properly. Okay, so the really big question that got me excited about doing this work when I first started in this field was that abnormal nuclear structure is a hallmark of human disease, abnormal nuclear mechanics and structure. And so this is true across not all diseases, but a little bit of every part of the disease spectrum. This includes aging, muscular dystrophy, heart disease, diabetes, atherosclerosis. Neurological defects or neurological problems have started to come on in the last 5 or 10 years as being nuclear morphology driven as well, or at least there are effects there. And a large subset of cancers, including breast, renal, lung, cervical, leukemia, and prostate. And I'm sure that maybe more are being added to this list, or that this list is not fully complete of everything. What's really interesting is you can see here that these abnormal shapes come in lots of different flavors. We mostly in our lab study nuclear blebs, which happen in progeria, this advanced aging disease. And another example of that is that increased bleb formation in prostate cancer determines its disease progression. So let's take a step back. We've known about abnormal nuclear morphology associated with human disease since the advent of the microscope. Since the Leeuwenhoek microscope in the 1880s, we started to look at healthy tissue, and we saw that they were ellipsoidal or circular shaped. And then we would look at diseased tissue from individuals, and we would see that the nuclei were abnormally shaped. Fast forward to the 1940s, where the test of the pap smear was invented at NYU. This took cervical cells and looked at cervical cells and determined if they were large and abnormally shaped. If they were, then that individual had cancer. How large and how abnormally shaped told you how aggressive that cancer was. So we've known about this for over 100 years. We've been using it for about 80 years to prognose and diagnose diseases and cancers so that we can treat them. People survive more from cervical cancer because we have this great prognostic and diagnostic measure. But we still don't understand why the nucleus deforms in the first place and what the causes and consequences are. And so we need to deal with that. Okay, so let's address the consequences of loss of abnormal nuclear morphology. Why should you care about a nucleus losing its shape? And the reality is that loss of shape creates high curvature. Here, what we're showing is bleb formation in this movie that's playing. This is NLS GFP. It's housed inside the nucleus, but upon formation of a bleb, the nucleus also ruptures, and you see that NLS spilling out into the cytoplasm. The nucleus does a good job of repairing this over about 10 minutes, but if that bleb, which usually persists, ruptures over and over again. And obviously, your nucleus, its main job is to act as a compartment to separate itself from the cytoplasm. So this rupture causes dysfunction through loss of this compartmentalization. So some of those big things are changes in transcription, increased DNA damage, and also changes in cell cycle control, right? And so when we entered the field, the problem that existed was, how do we know what determines nuclear shape maintenance, right? Because there's both chromatin filling the interior here and lamins at the periphery, and both of these are well-known mechanical components, but the field had been unable to separate their roles. So when I entered the field, I thought, okay, so maybe I can develop a new force measurement procedure that might be able to get at this question of how to separate chromatin and lamins. So we developed this dual pipette micromanipulation. And what you see here on the left is isolation of a living cell. So we're using a detergent to break open the plasma membrane. We fish out the nucleus and we put it between two micropipettes. We can then pull with one pipette and watch deflection of the other one. The left one, or the force pipette, has a pre-measured bending constant before this experiment. And so how far it moves, we can back out to get force. So then we basically can get a force versus extension measurement, which basically gives you the spring-like properties of how strong the nucleus is. When we did this, we got lucky, to be completely honest with you, and we were able to, for the first time, separate the roles of the major mechanical components of the nucleus, chromatin and lamins. What we found is that chromatin dominates the short extension force response, up to about 3 microns of extension, and lamins come on at longer extensions to create a strain stiffening effect. Now, the first question would be, does this agree with biology? And in fact, this agrees really nicely with biology. All of your cells, all of your nuclei, have chromatin inside them. But not all of your nuclei have high levels of lamins, specifically lamin A, which is the major mechanical component of the lamina. And so lamin A comes on during differentiation, and lamin A is upregulated in mechanically demanding environments like your muscles, your bones, and your cartilage. And so this actually makes complete sense. All of your nuclei have chromatin to put up with the base level of forces that they should see. And then when they differentiate into mechanically demanding environments, they upregulate lamins, which are mechanoresponsive, which if they feel force, they will sort of upregulate themselves in their incorporation in the lamina. And now you've put on a protective coating for anything that's going to see high deformations. It's also worthy to say that lamins are also very important for cell migration, in which you're going to see these high deformation regimes. Now we go back to the field where everyone thought that lamins were sort of responsible for nuclear morphology. And so then here's some of the core data, apologies. Here showing that when we decompact the chromatin, here's our wild type or untreated in black and gray. When we treat with VPA, a histone deacetylase inhibitor, to increase levels of euchromatin, we can decrease the short extension force response, but the strain stiffening remains. However, if we knock down lamin AC, we can see that the short extension remains the same, which is due to chromatin. And now actually, instead of strain stiffening, you get strain thinning, right? And so now you're literally pulling apart the nucleus in these high deformation regimes. Okay, so how should you think about chromatin and lamins interacting? And so if there's one thing you can take away from this talk, I hope it's this very basic model for how to think about the roles of chromatin and lamins in nuclear mechanics. Lamins, from Ohat Medalia's measurements, are actually really easy to bend, but hard to stretch. And that's a lot like a rubber band. It's really easy to bend from a circle, but then it's really hard to stretch once it's aligned along the tension axis. Then, when you add chromatin to the interior, what you can see now is that the 3D polymer of chromatin resists those initial deformations, and that chromatin helps bend the lamins faster to get them to stretch faster as well. Okay, so when we were entering this field, everyone thought lamins were very responsible for most of the behaviors of nuclear morphology and integrity. And so we depleted histone modifications to modify basically between these heterochromatin and euchromatin. And we showed that essentially chromatin independent of lamins can determine nuclear morphology, right? And so we've been able to sort of dial this up and down and in many different ways to show that this is in fact true. And the idea here is that if you increase levels of euchromatin, you decompact the chromatin and it's weaker, and then it succumbs to nuclear blebbing and rupture. But if you compact the chromatin by making it more heterochromatic, it's then stronger and it maintains shape. In this modeling that we did, so that paper and rubber band model is actually backed by rigorous physical modeling done by Ed Bannigan. When we built this model of chromatin and lamins, one thing that came out of it that was interesting is that, and I don't maybe think that this is actually interesting, but you kind of expect it, is that you need connections between the chromatin and you need connections between the lamina. And so we hypothesized those here. So chromatin is the polymer. I know it's hard to see, but there are these little green links, which are chromatin-chromatin interactions. There are blue linkages from the chromatin to the lamina, and those are chromatin-lamin interactions, and then the lamina. So you have 4 major mechanical components of the nucleus. While there are many chromatin to lamin linkers, we haven't quite figured out a way or one to determine that is a legit thing we find. There are many chromatin interactors that are actually quite big and responsible. And so in the years following, in 2021, we were able to show HP1 alpha is a mechanical chromatin-chromatin crosslinker, and this is a local crosslinker. And then working with Joop Dekker's group at UMass Med, we then also showed that high C interactions, which occur about every 20 kb throughout the genome, are also major mechanical contributors, right? So the idea here is that lamins are important for chaining nuclear rigidity and mechanics. Chromatin through histone modification states determining their compaction are very important for mechanics and maintaining morphology. And these chromatin-chromatin interactions are also another major contributor to maintaining nuclear mechanics and shape. Okay, so what I haven't told you about is that there's actually this, so who's the antagonistic component? Who are chromatin and lamins resistant? And there's a lot of work done by many people. I can think of Emily Hatch has been one of the big people in the field, showing that actin confinement here on the left was a major contributor to the antagonism of nuclear shape stability. We recently did a paper that was published in MBOC showing that actin contraction is as important, if not maybe more important than actin confinement, and is actually can behave independent of actin confinement, right? And so this is phosphorylation of myosin light chain. So this is actomyosin contraction. And that is also an important antagonistic force for deforming the nucleus and forming these blebs. And that can be shown here. When we increase actin contraction, we can increase nuclear blebbing levels in wild type. For some reason, our VPA chromatin decompaction control doesn't respond as well. And then in a lamin B1 loss, which is known to cause increased levels of nuclear blebbing, it can increase it as well. The big thing is when we knock out actomyosin contraction, you maintain normal levels of actin confinement, you absolutely suppress nuclear blebbing quite drastically across all scales. Okay, so then what is a nuclear bleb? And this just gives me a chance to sort of plug recent work that we've done with Vadim Bachman's group in collaboration. And what we found was that, in fact, chromatin and loss of chromatin density in the nuclear bleb is the best marker for a nuclear bleb. We rigorously rooted out the difference between a nuclear bleb, which is still connected to the nuclear body, and micronuclei. And so I suggest you go look at this paper, Bunner and Prince et al., recently published in JCS. And then we also did this alongside Vadim Bachman's group using partial wave spectroscopy as another way. So here you were using immunofluorescence or fluorescence staining. And when we use partial wave spectroscopy, we see a secondary technique give us the same thing, which is a decrease in chromatin density in nuclear bleb. And we had companion papers published alongside. So here's our paper and here's Vadim's paper. And we worked together on these two papers and they were published side by side with an article highlighting them. And so it's really cool that we now have a stable marker. In the past, people thought lamins were a good marker for nuclear blebs, and we showed that that's actually not true. Then that should bait out of you, why do lamins not label a nuclear bleb in a meaningful way? Well, they do, but the idea is slightly different. So we just put on bioRxiv our follow-up story to this, which is Chu and Lang et al., which is that lamin B1 loss in nuclear blebs is completely rupture dependent. So a bleb, like here shown, has not yet ruptured. But after nuclear rupture is when you lose lamin B1. What we also found was a very interesting thing was that DNA damage, which is associated with bleb nuclei, is actually not dependent on rupture. And so I invite you to, in your time, or if you're interested, go look up these papers. Okay, so getting back to sort of the main point for why you should care about it. Nuclear blebbing gives a high curvature region, which we think then creates this increased Laplace pressure, where the pressure on the inside is greater than the outside, causing nuclear rupture. And this disrupts major functions of the cell by disrupting transcription, cell cycle control, and DNA damage. And this has been reported by everyone in the field in a really consistent manner, right? And so the idea is this partially responsible for causing dysfunction and disease? And essentially, the answer we think is yes, right? These abnormal nuclear morphologies are essentially causing dysfunction, which might not be it's at least a contributor to the dysfunction inside of the disease state. Then we asked the next really important question, which was, what would be really bad is if the nuclear dysfunction that is caused by a nuclear bleb and nuclear deformations rupturing, what if that dysfunction fed back and caused a greater loss of nuclear mechanics, shape, and integrity? Then you would basically be in a doom spiral where you'd be progressing the disease as these things fed back to each other in a really, to cause a negative way. We say negative feedback in the meaning that this is bad, not necessarily as negative feedback that it stops it, but this is driving you down a path of greater dysfunction, greater disease. And essentially the story I'm going to tell you about today is that transcription plays a role, or transcription activity plays a role in changing the nuclear morphology and rupture rate. We also just published recently in MBOC that increased DNA damage causes loss of nuclear mechanics through loss of constitutive heterochromatin. And so then what you get is a nuclear bleb forms, you get increased DNA damage. That increased DNA damage could then feed back to then decrease heterochromatin levels more, which would then cause more blebs and more ruptures, which would then cause more DNA damage. And you can see how we're cycling here. Speaking of cycling, we also have a paper in preparation that we hope to put on bioRxiv maybe in the next month or two about how cell cycle control over nuclear mechanics and actin mechanics, this force balance between the nucleus, resisting actin confinement and contraction. And we find some really interesting things inside that. So please be on the lookout for that in the future. Okay, so I'll circle back to our main story, which is transcription. So let me be very clear. When I say transcription, I don't actually mean transcription as in DNA to RNA to protein. What I'm really talking about is transcription as an activity, as a physical activity. If you think of, and I'll sort of try to make it, if I'm RNA-Pol2 and I have a rope, If I pull in if I'm reading across this DNA, I'm going to pull in from the front, and I'm actually going to push out in the back. And RNA Pol2 is a strong motor. It's a motor as strong as our classic sort of motor proteins, thionine or myosin, right? And you don't even need those levels of force to get the things that we're talking about. But essentially, it's a motor that could be causing motion to happen inside the chromatin. There's a lot of support for this. Recently, there's been a lot of data supporting that transcription drives coherent chromatin motion. So coherent chromatin motion are 1 to 3 micron domains of chromatin moving in the same direction or moving in the same fashion. And here you can see Chavan et al. 2018 is one of the first to sort of dictate this. So with serum, with normal levels or high levels of transcription, you get a lot of coherent motion, and then you can suppress said coherent motion by not having serum, which decreases transcriptional activity, or you can have inhibitors of transcription in DRB or TPO, right? So the hypothesis that transcription-driven chromatin motion could push against the nucleus to deform. So then the question becomes, is this even like possible, right? So the idea here on the left is that you have coherent chromatin motion that could be driving, sort of pushing out at the periphery. And when they do simulations of shape, they actually show that this is possible, that if you put in a motor of undefined specificity, that motor could drive deformations of the nucleus. And this is Liu et al. 2001. So there's some basis for this in the modeling, at least. And then you look in the literature for maybe disease implications, and what you find is what I showed you earlier, which is that you can actually increase nuclear blebbing in prostate cancer cells is directly correlated with disease progression. You can also feed prostate cancer cells DHT or testosterone to activate the androgen receptor and then to drive up nuclear blebbing. And that's something we'll talk about later. So there are implications here. Okay, so we want to just start from the base. So there's the beginning of our story. Can we modulate transcription activity simply by suppressing it? And so we have here our untreated. We have VPA, which is our sort of positive control for decompacting the chromatin to induce greater forms of nuclear blebbing. When you decompact the chromatin, you create more euchromatin, and in fact, you increase levels of transcriptional activity as measured by RNA via E. You can also look at transcription activity via serine 5 and serine 2 phosphorylation. And so that tells you both initiation and elongation. And you can see these levels drop off drastically whenever we treat with alpha-minitin, which is a known molecule that binds to RNA Pol2 and degrades it over time. So all the data you're going to see is 24 hours with AAM, which I know is a long time. Hang in there, we're going to get through it, and you're going to see that there's a lot of data to back up. So when we start, we wanted to just ask the basic question. If you drove down transcriptional activity by trying to inhibit RNA Pol2, could you suppress nuclear blebbing? And the answer is yes. Then we ask the question, this is done in MEF cells, which is one of our standard cell lines. Then we ask the question, is this a phenomena that can be seen across multiple cell lines? And in fact, yes, in HT1080 is another cell line that we work with. You can again take this increase that you see from chromatin decompaction, and you can completely suppress it by inhibiting transcription with alpha-minitin. Then you would ask the question, well, is alpha-minitin, this really strong degradation of RNA Pol2, is this what you need to decrease nuclear blebbing? And in fact, no, you can inhibit initiation with triptolide, you can inhibit elongation with flavopiridol, or you can do a very fast inhibition with actomyosin D. And in fact, many transcription inhibitors give you the same result, which is that you inhibit nuclear blebbing. Then we ask the question, is this just a VPA-specific effect? So then we have our sort of, this is increased euchromatin. DSNP is for decreased heterochromatin, loss of lamin B1, and lamin A knockdown. All of these give you increased levels of nuclear blebbing, and all of these are suppressed when you inhibit transcriptional activity. Okay, so the question is changing transcriptional activity, just changing something we already know contributes to nuclear mechanics? And our list here out to the side is chromatin compaction rigidity, lamin amount and rigidity, actin confinement, and actin contraction. Okay, so then we needed to go through, is this a new mechanism or is it just something we already know? When we looked at chromatin compaction, what we saw was in fact not a rescue of of increased levels of euchromatin caused by VPA. In fact, actually levels of euchromatin even went up higher. And then when we did nuclear mechanics, I think this is really what blew us away. We saw no change in the nuclear mechanics. As you can see here, we have our untreated. When we treat with VPA, where we decompact the chromatin, the nucleus gets weaker. And when we have alpha-minitin and we have VPA, it's also weaker relative to the alpha-minitin, right? And these are not statistically significantly different. So we have the first treatment, at least in our knowledge, or at least one of the first, which is not changing mechanics. It's not changing chromatin mechanics. The question is it changing lamina mechanics or actin mechanics? But it's not changing chromatin mechanics. It's doing something else. Is it changing lamina mechanics? The answer is no. There's still normal levels of lamin A. We can look at this lamin A strain stiffening regime from our micromanipulation measurements, force measurements, and we see that those don't change either. I will mention that we do see these invaginations, which we actually think are the effect of if you have less chromatin motion, you have less pushing against the periphery, and so sometimes you get these invaginations. Then we asked, did actin confinement change? And what's really cool is in VPA, when the nucleus gets weaker due to chromatin compaction, because its nuclear height collapses, right, from here to here, because now it's softer, right? Your hand smashing the stress ball, it now smashes it more if the stress ball is weaker. That remains when we treat with alpha-minitin. So actin confinement is not modulated by this. And then actin contraction, which we showed you in Pha et al., is very important for nuclear blebbing. We also show here that when we treat with alpha-minitin, we do not affect that either. So what we've found is that the classical view of nuclear blebbing, this force balance between actin antagonism and nuclear resistance, is insufficient to understand how nuclear blebbing occurs. And so now, instead of just having this top model, what we have is added in here that chromatin motion via RNA Pol2 transcriptional activity, not transcription itself, but transcriptional activity, RNA Pol2 acting as a motor to move chromatin, is necessary to form nuclear blebs, as shown here. So now there's sort of two components that need to come together. If this was true, then maybe we would see some sort of intermediate phenotype. And in fact, we do. When we have normal levels of transcription activity in the top here, you form a bleb. The bleb ruptures very soon after. At 10 minutes later, that nucleus will heal the nuclear envelope rupture, and that NLS-GFP will reincorporate, and that bleb remains. So in many cases, what happens is a bleb forms and it stabilizes. And we have other stories we're working on in the lab where we think this persists for a very long time. When we inhibit transcription, when we stop transcriptional activity, we see blebs that are able to form rupture, but then upon NLS-GFP reincorporating into the nucleus, that bleb is now gone. And so what we move from is when transcription is active, we see bleb stabilization, and when transcription is inactive, we see those blebs not stabilize. They form and then they go away. They're very transient. When we looked at our treatments of these inhibitors over this long period of time, what we saw was that bleb reabsorption went up drastically, right? So not only do you need it for initial formation, you need it for stabilization immediately after formation, and you need it for stabilization after it's already been around for a long time. Then we looked at transcription initiation here in purple and elongation in gray. And what I told you before is that the DNA is decreased inside of the bleb, right? And you can see that here. In untreated, we get about a 60% of its normal level, right? So a third decrease. What we found really interesting was that transcription inhibition, or transcription initiation, was more enriched in the nuclear bleb, which is a great example here, than elongation or DNA. And that was consistent across all conditions. And so this really suggests that something specific with transcription initiation is really important for forming the bleb and stabilizing the bleb. We're still unsure what that is, but it's an interesting tidbit to share. So then we asked, is chromatin motion sufficient to create nuclear deformations in a rigorous model? So we rebuilt this model using Sartak Gupta ran this research out of Jen Schwartz's lab with help of Ed Bannigan and Alison Pattison. And we recreated the mechanical model, chromatin interactions in red, chromatin lamin linkages in green, the lamina in purple. And these mechanics models are actually really sophisticated. This is like the 5th or 6th round of publishing said model as it gets more complicated or as we do different things with it. And so there are lots of papers backing up sort of this mechanic. Now we're looking at its morphology. And what we do is we drop in a repulsive motor and see if that's sufficient to create deformations. And so what we simply did is modify the number of motors. And when you go from low motors to high motors, or from low transcriptional activity to high transcriptional activity, you increase the deformations. We call these bulges because this is in simulation, so they are not akin to nuclear blebs, but they are the modeling similarity to it. We then ask the question, is this due to chromatin motion, right? And we were able to go in and look at chromatin trajectories from our model. And what we found was that underlying these deformations was, in fact, coherent chromatin motion that was already talked about from Hythe and Shaban's group. Alexandria Zadovska's been talking about this. I'm going to forget one of these names, and Pierre Vivi. I'm sorry for the last one that I'm missing. I'm so sorry to. But a lot of people are seeing this behavior. So we have data showing that transcriptional activity is necessary for blood formation. We have a mechanical model backing up that our hypothesis for chromatin motion has legitimate data supporting it. And so that is the end of our published story. We've obviously continued to try to understand this data at a higher level. And so I'll give you a quick thing on this very boring slide, which is that we upset some people during this process by saying transcription. And many people did not buy it. And so we needed to start to adapt to the way that our colleagues were receiving this information. They were really upset that we were inhibiting transcription over 24 hours, and that was causing a lot of off-target effects. So what we've really done is gone out to try to lower the off-target effects and do this in multiple ways, and especially do these in ways that are disease relevant. So I'd love to talk about those now. So the fastest way to get rid of RNA Pol2 is to degrade it. So these are amazing RNA Pol2 degron constructs, I think made by the Kanamaki lab, and we got them, and we were able to degrade these in about 6 hours. And so this is really what we're trying to do, is degrade RNA Pol2 as fast as we can to minimize the impact on transcription. Then we thought it would be nice if we could downregulate it and then reactivate it, so sort of dial it down and dial it back up. And there's actually really great, so modulate it. And there's really great data in the literature about how serum starvation will turn down transcriptional activity, and serum stimulation over a very short period of time will turn that activity back. Finally, we want to do some disease-relevant increase in transcriptional activity. So one big feedback we got was, you're changing the whole transcriptional activity. What if you did something very specific? And we have a model for that, which is testosterone, the androgen receptor, androgen responsive genes, and prostate cancer cell models, which increase levels of nuclear blebbing. So now I'm going to tell you about each of those. So first, we take this RNA Pol2 AID GFP degron construct, and we're able to add auxin. And at about 6 hours, we can almost completely deplete it, going from very clear, obvious presence of RNA Pol2 labeled with GFP to nothing. And when we did this over just 6 hours, we were able to suppress nuclear blebbing in this HCT116 cell line. So now we're into like our 3rd or 4th cell line that we're doing this in. I think we have even more than that I just haven't shown you the data for. All right, so we can do this quickly to try to alleviate off-target effects. Then we thought it would be great if we could use serum starvation over 72 hours to decrease levels of transcription, and then serum stimulation over about 3 hours. And so we wanted to be able to see how this would change initiation and elongation. And the long and the short is serum starvation is as good at turning down transcriptional activity, or at least the phosphorylation of RNA Pol2 for both initiation and elongation, as degradation, right? And again, it's not turning off transcription, it's turning it down to a really low level. So we're not degrading RNA Pol2, we're not turning it off completely, we're turning it down really low. And then when we feed serum stimulation to these starved cells, in about 3 hours, they can turn transcriptional activity back on. When we do this, what we see is a very clear recapitulation of the data I've been showing you throughout this entire talk, which is that we can go from bleb nuclei to suppressing that nuclei back to a normal shape when we serum starve over 72 hours. And here you can see we actually end up using the 0.5 to keep them the happiest. And what you actually see here is if you use 0.1, you can actually suppress nuclear blebbing in wild type. If you use anything 1%, 1%, 0.5% or 0.1%, you can really just suppress nuclear blebbing in our increased nuclear blebbing case of EPA. We then take these same cells and then we dump on serum, and in 2 hours, we can return nuclear blebbing levels, right? And so this is a really cool way of dialing it down and then dialing it back up. I don't have time to show you all the data, but none of the other stuff that we're interested in, chromatin compaction, lamin levels, lamin rigidity, actin confinement and actin contraction are not significantly modulated when you're doing this. Then we wanted to start to look at motion. We are actually doing chromatin motion in a very different way, but we're still trying to figure it out. But this is just a really nice initial movie we had. This is H2B. We're looking at H2B low density regions as a bleb forms. And what you can see here is there's 2 dots here that get pulled into the bleb and then end up at the edge of the bleb. And so this was just an initial look showing that chromatin is in fact flowing or pushing against the nuclear bleb. You can see that this starts to move even before the bleb starts to form. And so we've started to ask questions about, does chromatin motion does chromatin motion underlie these changes in transcriptional activity? And so now we're doing other things that we just don't have the data to show you, but we hope to have it ready to show to you. Maybe in 2 months, we'd like to submit at least maybe a bioarticle. Okay, so for the last thing we wanted to talk about disease relevance. So we're turning up, we're knocking it, we're degrading RNA Pol2, we're inhibiting it with these really strong inhibitors. We're modulating it by serum starvation and stimulation. Like, we're doing a lot of different things, but we're hitting the whole apparatus. What if there was a specific pathway that drives transcriptional activity that could underlie a disease phenotype? And so these LNCAP prostate cancer cells, along with PC3 and DU145 and their wild type correlates, provide a really cool model here. So there's this Helfand et al. 2012 paper that really nicely showed that you could feed prostate cancer cells, these LNCAP early stage cancerous prostate cells, DHT, so testosterone. This would activate the androgen receptor, and androgen responsive gene activation would occur, and you would increase levels of nuclear blebbing. This is important because in tissue samples, these are tissue samples taken from individuals with prostate cancer. You can see that in normal healthy prostate tissue, there's very low levels of nuclear blebbing. But then when you go from sort of early grade to more advanced grade prostate cancer, measured by Gleason score here, you can see that levels of nuclear blebbing increase during this time. So we were able to recapitulate this phenomena where we treated cells with testosterone, and we increased levels of nuclear blebbing in these LNCAP cells. We then did our transcription inhibitions and suppressed that behavior to show that it is in fact responsible for this. We also inhibited the androgen receptor, and it blocked this behavior as well, showing that it's specifically DHT binding to the androgen receptor. And we'd like to continue this work. This is a continual thing that we're ready to share all the data, but we really wanted to show you some of the data because it really drives our point. And the reason you should care about nuclear blebbing is the same thing I told you at the beginning of the talk, that nuclear blebs are sources of dysfunction. And the most easy dysfunction for us to measure across the field is increased DNA damage via gamma H2AX or 53BP1. And here, as we've shown in many other cell lines, normally shaped cells have very normal levels or very low levels, relatively speaking, of DNA damage. But in bleb nuclei, you increase levels of DNA damage. I should actually be frank that this is not even a great example. In these LNCAP prostate cancer cells, usually the bleb is completely soaked in gamma H2AX. That bleb region becomes what we think is possibly a hot spot for DNA damage. And we hypothesize, as I think Louis has talked about in the past, that this This area is unique, and in fact, there's increased transposon activity, which is directly linked with disease progression in prostate cancer. And so we have this more advanced idea that we already know nuclear blebs cause dysfunction, but now we have the secondary idea to track inside of a specific model for prostate cancer, that it's possible that the bleb becomes a hotspot for DNA damage, and then DNA translocations, which are known to increase in prostate cancer. And so this is some of the stuff we hope to track down in the future. But the big take home from this is you're not activating the whole genome. You're not turning transcription up and down drastically across everything. This appears to be an activation of a single pathway. Obviously, we need more data to prove this, but it appears at the initial step that you can activate a single pathway, drive up transcriptional activity, and increase nuclear blebbing and ruptures. And this leads us to the idea that maybe things like c-MYC, KRAS, HRAS could be turning on to upregulate transcription across many cancers and pushing them over the edge to generate more nuclear deformations that lead to more dysfunction that then lead to disease severity. Oh, sorry, skipped. So the conclusions are that transcriptional activity modulates nuclear blebbing, and this nuclear blebbing is important because it causes nuclear dysfunction through rupture and through this unique location of the nuclear bleb. This behavior is a new addition, right? It's independent of the established nuclear rigidity and actin antagonism force balance. It's a separate thing. We hypothesize that chromatin motion is the plausible mechanism for nuclear bleb formation and stabilization, and we're trying to gather data towards this, but we already have a mechanical model that suggests that this is plausible. And finally, that transcriptional activity in disease can drive nuclear blebbing, leading to more dysfunction, and it can do so in a pathway-specific manner. You do not have to change drastic levels of transcriptional activity. Maybe a single pathway or maybe even single transcriptional activators would be sufficient to push this over. And so, in fact, what we're interested in is that these RNA Pol2 motors with the chromatin linked together push it in mass against the periphery to help form these nuclear blebs. And without their work, nuclear blebs cannot form and not cause the dysfunction that's associated. All right, so I'd like to thank everyone associated with this work. Isabel was the leader of the project I talked about, and everyone who was on the paper. Kelsey Prince is leading the charge for the new transcriptional activity data that we hope to have out in 2 to 3 months. We worked really strongly with Ed Bannigan, who is who's built the mechanical model. We worked with Jen Schwartz, Sartak Gupta, and Alison Pattison to develop this model into a morphology model with these motors. We have standing collaboration with Bob Goldman and Team Adam. We want to thank Karen Reddy for some of the cells that she gave us. And then, of course, the team at Northwestern, specifically, we work a lot with Lou inside of the Deems lab, and Nicholas has helped us out with this. And then here's all of the people in my lab who are amazing and really make it what it is. And we want to thank our funding from the NIH and the 4D. And now we'll take any questions. Thank you for your time.
Thank you so much. That was fantastic. As I said, if you have questions, feel free to just turn your mic on or type them into the chat.
Hey, it's good to see you.
Hey.
So I actually had a couple of questions. Well, first I'm going to ask, did you, since you're seeing androgen stimulation or testosterone stimulation is driving it, you know, like the list of androgen responsive genes is not very large. So are you going to do FISH and like nail down that they're going into the blebs or near the blebs?
I think that's what we'd like to do. Right now, we're working hard on the chromatin motion part of it, but I think when we have more time to circle back, this Helfand et al. Paper actually did this. It looked for these androgen responsive genes and found them in the bleb more often than in the body. And so someone has done that work. I think we want to do it again. It would be great work to have a second paper showing that. And I think we want to understand sort of like where those genes are before, during, and after the stimulation.
Yeah, I think I need to get you like meeting with Kyle McQuarrie, who's doing this kind of on the other end in like pediatric cancer. But I think like if you guys approach it from two different cancers, you're gonna show something amazing. Then the other thing I think that you said that was actually quite surprising was that chromatin went up when you inhibited transcription. You just like went
Yeah.
straight past that, hoping that nobody would ask you questions, but
Yeah, I don't know why that happened, to be quite honest. I don't know if this is I don't know. Yeah, I don't know why this is happening. I could probably make some guesses, but I don't think I would have any data to back up. I think what we were really worried about was that if we inhibited transcription, that chromatin would essentially like collapse on itself without RNA Pol 2 actively being associated with it. And so we were really worried about maybe some dynamics in which chromatin would dissipate, and we didn't see that. This increase is interesting, but we're just happy it didn't go in the opposite direction.
yeah, I think you and I should meet at some point and like we can cross notes, but I think there's more to chromatin and like its tertiary structure that probably explains why you're seeing it go up.
Right. Okay.
I have one more question unless anybody else wanted to ask stuff, but the motion, it looked like transcription was trying to go outside.
Yeah, so what I can tell you is that we do see transcriptional when we modify transcriptional activity by serum starvation and stimulation, we see chromatin motion increase, and we do see it sort of pressing against the periphery, right? We're starting to ask questions about, is it just motion at the periphery? Is it also motion internally? And that's, I think your eye really associates with stuff at the periphery a lot more than the interior. And so we're doing measurements now to make sure that the interior is also fluctuating at but yes, we really do see that the chromatin appears to be pushing against the periphery, causing these deformations. Or there's more chromatin activity in serum stimulation, less in serum starvation. And when there is more activity, we see it sort of colliding with the periphery.
I think in your micro domain, like just trajectories, you can see the arrows. Like, I've talked about this before, but like there's that region where when you inhibit heterochromatin, you get like this like intermediate ring of chromatin that pops up, and that was where you actually kind of like, if you like could superimpose the two on each other, it was like that second halo is where your outward vectors seem to be coming from. And I thought that was really interesting.
Yeah, I think it's worth mentioning that like that ring of chromatin that happens when you modify histone modifications, we don't see that happening when we disrupt lamin B1, and we don't see that happening when we disrupt lamin A via knockdown. And so having these joint sort of BPA, chromatin, DSNAP loss of heterochromatin, LB1 knockout, and LAKD knockdown, having this battery of different perturbations to look at, I think is really strong for us to make sure that we're not getting too engaged in one specific behavior, right? And so while I think that behavior is interesting, it doesn't appear to be necessary for us. I think there's some stuff in the chat. I don't know if anyone wants to ask. Sorry, go ahead.
Sorry, I just wanted to piggyback off that, Dr. Stevens. I'm Cody Dutton. I work in the Dean's lab as well. So I think that this idea to me makes sense in terms of like cancer cells, and if you have increased transcription, increased blebbing and disease, but with aging, it almost seems like in senescent cells or cells age, my understanding is that there's a little bit like we decrease the amount of division, we potentially slow down some of this transcriptional activity, but we still see increased blebbing in, say, senescent cells. So I was wondering if you could speak on how we could rationalize this idea of decreased transcriptional activity, but also still have the increase in blebbing that's associated with senescence.
Yeah, I think that's a really interesting point. During senescence, you're also going to get drastic changes in histone modification levels, and you're going to get drastic changes in lamin composition. And so we sort of think about this as like maybe not an all or nothing, but a spectrum, right? And so the question is, so maybe I should answer it help in this way. These LNCAP cells, we went back and looked in the literature. Someone else has measured LNCAP cells. LNCAP cells are mechanically weaker than their wild type correlate. So they're already mechanically poised to form a nuclear bleb, right? They just need a little bit of transcription input. And that's a single pathway, testosterone to androgen receptor, right? And so if you think about it as a sort of like spectrum, you might be downregulating transcription, but you're really going to hammer the mechanics of the nucleus in these senescent cells. And
It's almost
so
like a ratio. You want to balance transcription versus the mechanical activity, especially say lamins at the periphery.
right. So maybe if you have histone modifications that are going, maybe your chromatin compaction is not what it needs to be, and your transcription activity goes down, maybe you don't bleb anymore. But maybe if your histones are hit, euchromatin, heterochromatin, constitutive heterochromatin, facultative heterochromatin, lamin A to B composition, and then you downregulate transcription a little bit, it's not sufficient because it's already mechanically really disrupted.
Okay. That's interesting. That's a good point. Thank you.
It's almost like it integrates all the mechanical components together.
But I mean, it's a good point. We should think more about senescence. I think this popped up once and it hasn't been on our radar, so we really appreciate putting it back on our radar as something we should be considering. So thank you.
Thank you, and thank you for your thought.
So we've got a multi-part question in the chat. Do you want to maybe just read, since it's multi-part, what the best way to do that would be?
Sure, I'll read it out loud. So thank you for your very exciting, insightful talk. I have several questions. First, does the proximity of the transcriptionally active chromatin to the nuclear periphery play a role in the blebbing? This is a fantastic question. I think we would need to look into, we don't have this capability yet. We would need to know exactly what genes we were going to transcriptionally activate. So say if we knew where the androgen responsive genes were, if they were at the periphery or the interior, and then we watched whether they moved from the interior to the periphery, that's an interesting question for us to think about. What I can tell you is, you know, the more active chromosomes usually live on the interior, but in Bob Goldman's past studies, those more actively transcriptional euchromatic chromosomes end up in blebs more often. And so I think if you're at the periphery and you're mostly heterochromatic, you might maintain your compaction and your stability to stay at the periphery, and that it's actually these euchromatic or transcriptionally active sort of more fluid chromatin that allows itself to flow and collide against the periphery. That's just my guess. Considering that chromatin at the nuclear periphery is mostly transcriptionally inactive, do you think that this could affect the locals? The question was, considering chromatin at the nuclear periphery is mostly transcriptionally inactive with the exception of chromatin around nuclear pores, do you think this effect could be localized to the inter-lad domains? We do not have a current way to measure at inter-lad domains. This is an interesting idea. I sort of still think that chromatin that's at the periphery, or sorry, chromatin at the interior that is transcriptionally active is actually moving quite, is maybe pushing against the heterochromatin at the periphery and trying to work its way through this dense layer of chromatin and lamins to push out, right? And so the idea is of a frustrated polymer. So if you have two polymers and this polymer opens and fluctuates more, it wants to gap the other one to escape. And so that's sort of how I would think about it. We don't have any measurements to back. Do you think nuclear pores can play a role in this process? Not that I know of. This is a great question. It's possibly they could, just through their interaction with things that are usually transcriptionally active, want to be at the nuclear pore to export their mRNA after they've made it, but nothing that we've seen directly. There's no direct evidence of how nuclear pores can contribute to this process, but it's definitely something we'll think about. Also, is transcriptionally active chromatin the one that drives deformation, the one that ends up in the blebs? Yes, so we see transcriptionally active chromosomes end up in the blebs. That's Bob Goldman's data. In the Helfand et al. Paper, the androgen responsive genes in those LNCAP cells that bleb more, those androgen responsive genes end up in the bleb more often. And so in general, we find euchromatin enriched in the bleb. And so that sort of suggests heterochromatin, we're still figuring out which types of heterochromatin, doesn't move into the bleb as often. And euchromatin or open chromatin, that's likely being transcribed at some level or capable of being transcribed, is what usually ends up in the bleb, right? And the bleb, of course, is really decreased density chromatin, We also have another question from chat, unless anyone wants to jump in. While blebbing is related to disease, but transcription leads to more blebbing, I'm assuming that healthy cells would still have some level of transcription compared to nothing at all. So is blebbing a natural occurrence, but it's the repetition that causes disease? We see blebbing at very low rates in our wild types and in healthy tissue. Blebbing itself is sort of a stochastic event, so only a subpopulation will bleb. Whether I can say healthy tissue blebs like 0, I don't really have data for that, but it blebs at a very low rate. We also see that those blebs tend to not persist very long, whereas with perturbations, they persist for a lot longer. That's data I didn't show you, but we do have. I do think that it's essentially the idea that you mentioned at the end, that blebbing does occur, but very, very rarely in very, very few cells. It's the idea that blebbing increases, and then that causes dysfunction, which can feed back and cause more nuclear blebbing. And then that sort of starts to spread, right? Throughout the population. If that's a dividing cell set, then that cell that had a problem is now going to become 2, 4, 8. Or it may suggest that the entire sort of subpopulation is descending, and then the blebbing helps them essentially descend faster, right? So I think it is not about the one-offs, but about the repetition of it happening over and over again, and then it happening over and over again in more cell lines. And then here, transcription would be important because maybe a bleb forms in wild type. We've actually started to see blebs form on really short timescales that we didn't think they could form on before. In transcription inhibited instances, they form, they rupture, and they go back. And then the question is, does a single rupture even matter? Does that cause dysfunction? And what we would say is that a persistent bleb is really what causes the dysfunction. Maybe not even the rupture, just the idea that you're taking a portion of your DNA and sticking it in a very different compartment, and it lives there for a long time, and it occurs this insult over and over again that I think really causes the problem.
Yeah.
Thank you.
I think if you think about it, like our tissues aren't like a uniform progression, right? Like, so like if you have like a highly correlated situation, right? Like you have like active inflammation and like all the mechanics of that tissue are messed up, and then the ions are messed up in that tissue, and then the transcriptional signals are really high for repair, then, you know, you're doing like a massive disruptive calculation all at once. And so instead of like thinking about like, you know, like you said, the one-offs, it's like what happens when you have highly correlated events, and then that's going to drive the entire homeostasis for millions of cells out of their like normal boundary. And now you're starting to get to hysteresis of like cancer or aging or something like that.
Yeah, I mean, I like that. I like that the term of hysteresis. It's like you've gone down a path, but now you've gone too far that you're not going to be able to return to what you were before. And so it's going to accumulate over time. But it's about persistence, being in that for a persistent period of time. All right, thank you.
Okay. Any other questions? We're right at 1 o'clock. Impressive timing. Okay, so if no other questions, I think we can wrap things up. I'm just going to share my screen briefly so I can show everyone our upcoming seminar schedule. Thank you so much, Dr. Stevens, for doing this. Super talk, very interesting stuff. Glad we had some.
You want me to stop sharing?
Too.
You want me to go ahead and stop sharing?
And this will be up on our video archive, most likely later this evening. So thanks again for doing this. Let me just show everyone here's our upcoming talk schedule. Got some good ones coming up. So thanks to everyone for joining us. Have a great weekend, and we'll hope to see you in March for our next seminar. Thanks so much, everybody.